So we have covered pretty much the basics of cellular agriculture and precision fermentation. We are not going to repeat those basics, but what we are going to discuss today would be something that will enhance um, the understanding that you have had through the lectures in the past couple of days on how to produce them. So we know what cellular agriculture is, we know what precision fermentation is, we know what enzyme engineering is, system biology is, synthetic biology is. Now, you can implement those to tweak your hosts or even tweak your product or type of product that you are expecting after the fermentation process. But the question would be, what sort of reactor will you choose? And what are the different types of reactors that are available? Why we actually look into bioreactors in one way or other? And what would be the design principles that are involved when you are actually designing a bioreactor for a specific purpose? Now, before I start the lecture for today, I did receive a question with respect to the case study 2 that we were discussing yesterday. Do you all have any questions with respect to what you are going to do in your case study 1 or case study 2? What I have provided you with respect to the case studies was the product. What you have to do is go backward, develop the whole process. Okay? Now, the product that I have stated might not be the final product. It might be just the product that would have received from the industry, but it cannot be consumed. Let's say monolin cannot be consumed if it is not heat stable, right? Enzymes are also not heat stable. So the alpha amylase that you will produce, how do you want to sell it? If you want to sell it, do you want to encapsulate it? Do you don't want to encapsulate it? What would be the optimum way of producing the final product has to be decided by you, not me. Okay? Let's start the lecture for today. So we are going to start our conversation with bioreactor design and analysis. But before we get into the design and analysis part, we will talk a bit more about the bioreactors. We have covered majority part of the bioreactors, how they are implemented or how they are used with respect to your cellular agriculture and precision fermentation. But we'll we'll go through it again. So bioreactors, as you can see from the picture here, those are the huge ones, and all those pipes that you see here, those are the controls or any of the inlets that are providing the substrate inside the bioreactor. So it's a pretty complex unit. When you look at it, it's a very complex unit. When you operate it, it actually looks very complex, but it's a very simple system. In principle, it's a very simple system. What it does is very simple, but the way it has to be operated is very complex. And that's the reason why you have to have the engineering design principles involved here. And we need to understand how to design a bioreactor. Now, we are using enzymes, plants, and animal cells, or we are even using microbes as our host. Enzymes, if you're just providing a substrate, you put in the enzymes, you can get the substrate converted into the product directly. Plant and animal cells, we have already discussed multiple times. They can be used as a host to produce your product of interest. Microbes are widely used as a source, as a host to produce your product of interest. So then there are different types of products that we do develop. So there will be the bioconversion products where you have the substrate being converted into product. Biotransformation, where one substrate has been transformed into a product, and the recombinant products is what we have discussed as part of the precision fermentation, where we are developing the product using recombinant DNA technology. So we have genetically modified our host to produce the product of interest. So a bioreactor, by definition, is a device in which a substrate of low value is utilized by living cells or enzymes to generate products of higher value. Now, substrate of low value if you are using pure glucose or pure fructose or galactose, they are not cheap. They are abundant, but they are not cheap. When we do talk about low value substrates, we are thinking about the feedstock. So let's say if we are able to utilize the agricultural waste. So that's the reason why I give this particular definition there. But majority of the time, the final product that you're developing, that is of super high value. So that's why the substrate cost doesn't matter much. Even if you are using galactose, sucrose, fructose to produce your product, if the final product has a way much more higher market value, your substrate cost will not be of any concern. Correct? That's the same case that you will have in your case study 1. If the monolin is able to capture the whole market or the whole food industry market with respect to the sweetener part, that particular product is going to have a pretty high value, a market value. So your capital cost involved in terms of choosing the substrate might not be of concern to you. Okay, so be wise. When you're doing the techno-economic analysis, you would be able to figure it out. What should be your input, what should be your output, and how you are going to do that. Now, the bioreactors have been used to produce different types of products. Some of those that I have mentioned here. So, vitamin B12, those are produced through genetically enhanced microbes that develop it through one-step fermentation process using vegetable oil as a feedstock and sugar as a nutrient. We have genetically enhanced microbes producing baking enzymes used to enhance rising, strengthen dough, and prolong freshness. We have already discussed how the enzymes will basically provide a sensory aspect to our products, right? Microbes and fungi are genetically enhanced to produce enzymes that are added to as a brightening and cleaning agent. We did discuss about that. The protease enzymes and the lipase enzymes. The lipases are mostly used to remove grease. You would have heard about not natural, but biocompostable detergents. They're not the regular detergent, liquid detergents, but they are like compatible, more environmentally friendly. They actually have more of these enzymes, genetically enhanced enzymes or genetically produced enzymes. Now, bacillus microbes ferment corn sugar to make lactic acid, which is heated to create biodegradable polymers for woven fabrics, and these are the ones which are used to make diapers for kids, for babies. Now, this example, this particular slide shows different examples of fermentation system, like how fermentation has been used, but majority of the examples that I provided are more inclined towards a recombinant DNA technology, right? Always using a genetically modified organism to produce a product, because currently that's the norm. The regular fermentation, we are using Saccharomyces cerevisiae to convert barley into alcohol, right? Those will be the regular fermentation, the traditional fermentation. So traditional fermentation is still going on. They still use the same type of bioreactors. Even in there, you have to choose whether you're going to go for a specific bioreactor, which might be a batch reactor or a fed batch or a continuous stirred tank reactor or even a plug flow reactor. So there are different types of reactors that you can use. Now, what will you use for majority of your precision fermentation processes? That is what my interest was, and that's the reason why we have these examples. Okay. So biochemical synthesis versus chemical synthesis. Why biochemical synthesis is better is because it is mild reaction conditions. You are not using very harsh reaction conditions. If you remember from the slides of yesterday where we are producing the PTA, at that time we were looking at cobalt, manganese being used. Those are harsh chemicals which are basically being used. It has a very high regio and stereo selectivity, because if you're using enzymes, you have the capability of this. It's sustainable if you are using a sustainable feedstock. It has a better product yield. We did compare, if you remember, we did compare the PET with the conventional PET. Non-toxic and undesirable waste as byproducts. So They are non-toxic in nature, and the byproducts which are produced are not toxic either, and they can be used further for other purposes if need be. And it is more environment friendly. So the GHG emissions and all those things are reduced because we are changing the feedstock. By using a cheaper feedstock or a waste as a feedstock, you can reduce the overall GHG, and you can make it a very carbon neutral system. You know what carbon neutrality is? So let's say you have plants. Now you use the plants to make fiber. You use that fiber for making clothes. Now once the clothes, like the production of the clothes, the CO2 which is generated. So let's say this is the industry which is producing the clothes. They are generating the CO2. Now this CO2 is again used by the plant to grow, right? So basically it becomes a carbon neutral cycle. So that is what I am talking about. It's more environment friendly. High regio and stereoselectivity. If you're using enzymes have the lock and key functionality, right? So depending upon the active sites, it might, and the substrate that you are going to use, it will be very selective to that particular substrate. So if you are using multiple substrates, but you only want one substrate to be used, you can actually use that. So that's what it is trying to say. So you have the regional and the stereo selectivity.
Thank you, sir.
Now this is an example of a typical bioprocess that you can see. So you have your media and nutrients which are coming in through ultrafiltration into a bioreactor. You have water which is being added to the bioreactor through reverse osmosis. Air is being passed in to provide the oxygen to the bioreactor. And then you have, once the cells have grown to a specific concentration, they are moved into membrane filtration. Now, after the membrane filtration, some of that cell go back as cell recycle, or you can harvest the rest of the cells. Some you put back, some you reharvest, and whatever product you have had after the membrane filtration, you can do the downstream process, so the downstream processing. So this is your downstream processing. Now this is where the maximum cost is. Now just a quick question, why do we do a cell recycle? Any ideas?
Sir, when we operate at high dilution rate, we do not want our cell biomass to decrease in a bioreactor.
Okay. You have gone way much more in advance. We have not reached the dilution rate yet in our discussion, but to a certain extent, yes, but more of to maintain the same culture. You don't want to change the batch of your culture, right? You want to maintain the consistency. So what is the best example of cell recycle in your day-to-day life? Yeah, yogurt and curd, right, at home. So you keep the culture. After you have finished the yogurt, you keep a little bit of it so that you can reuse it. So that maintains the consistency. Cell recycle allows you to maintain that consistency. Okay. Oh, I could have asked another question. The media and the nutrients that are being added into the bioreactor, they go through ultrafiltration. Why? You need them to be clean, but you also need them to be sterile. Anything that is added into the bioreactor has to be sterile, except the cells. Okay, all the nutrients and the media that is added into the bioreactor at all times has to be sterile. It cannot have any contamination, because the moment you have a contamination, you're going to change the whole profile of your bioreactor inside, right? Now, this is a typical bioprocess. I'm not saying that this is the bioreactor design that we are following, but what type of bioreactor will this be? It has an input and it has an output. If it was a continuous input and output, it becomes a CSTR, a continuous stirred tank reactor, right? If it has only an input, it becomes a fed batch. If it has no input, no output, it becomes batch. right? Most of you have the background in biotechnology. You would have taken a course in bioprocess, so I know that you are aware of it. For those who don't, there are 3 types: batch, fed batch, continuous. So design of a bioprocess depends on a couple of things. The most important would be how fast will the process take place. So the reaction kinetics. What changes can be expected to occur? Like what would be the metabolic state? What would be the cell physiology that would be reached? And how will be the system be operated and controlled to provide the maximum yield and productivity? So what sort of design of reactor we are going to use and what sort of instrumentation and control we are going to have. So what would be the temperature profile? What would be the pH profile? How much oxygen is going to be purged? What would be the speed of the impeller within the bioreactor? All that has to be controlled. That comes under the topic of process control and instrumentation design. If you're looking into chemical engineering, that would be process control and instrumentation design. So the other aspect of the design of a bioprocess would be how will be the products be separated or recovered with maximum purity. So the downstream processing. Now we have discussed to a certain extent in detail about downstream processing. We did talk a little bit about the temperature and the pH yesterday when we were discussing about the enzymes and how they might impact the structure of the enzymes. So if let's say I am assuming that you are aware what a cell culture looks like. Okay, so if you have a flask, have you ever seen a flask with a cell in it? In the microbiology, if you have taken a course in microbiology, you would have seen that, right? Now, if I heat it, what will happen to the cells? They will die, right? So temperature, so the moment, let's say if the cell cultures were at 25 degrees Celsius, that's the normal room temperature right now, okay? So 25 degrees Celsius, which is a normal room temperature. If I slowly increase this temperature all the way to 70 degrees Celsius, will they start dying the moment I start increasing the temperature, or will they die after we have reached a specific threshold temperature? So once we have reached a threshold temperature, right? So temperature actually sometimes will promote cell growth. Eventually, once it has reached a threshold, it will start dying. So you will see that the cell will start growing, and this would be the temperature at which, or T max, that you can actually have. Now the reaction kinetics, like how fast will the process take place, it is very, very important. If you're looking from the chemical engineering perspective, the chemical reactors, you are using chemicals as your substrate, and the product that you get is also a chemical. You are providing a catalyst, you have to control the temperature and the pressure, everything. Now, some of the reactions are super fast, some of the reactions take time, right? And that's the reason why we add catalyst, so that we can increase the speed of the reaction. Then some of the reactions are exothermic, some of the reactions are endothermic, right? When they are exothermic, what does that mean? Thank you for the unity. I would prefer if one person can answer that.
Energy will be needed, endothermic.
So when you're releasing the energy into the system, that's exothermic. So that means you're increasing the temperature of the system. Am I correct? Endothermic, you are absorbing the energy. Does that mean that I'm reducing the temperature of the system? Yeah.
Exo.
Yes or no?
Exo will endo will take.
So if I'm doing an endothermic reaction, does the temperature of the system decreases? Thermodynamics 101. Tell me. What will happen to the entropy of the system in an endothermic reaction? It will reduce, right? The temperature might not. There are different exceptional cases, but temperature might not. Okay. So see, your thermodynamics is now involved down here. You have to have a background in thermodynamics if you want to do a bioreactor design. So an engineering background is very much required. Now, what changes can be expected to occur when we are looking at the metabolic state, the cell physiology? So, if we are aware of how which metabolic pathway is going to be followed by our cells, we will be able to provide them with the right nutrients which would be required for that metabolic pathway to get you the product of interest. Okay, so understanding that will also help. Understanding what is the growth rate or how quickly our cells will double, what would be the physiology, what would be the concentration of our cells later on will also help in designing the bioreactor. Understanding that the thing that I'm trying to state here is that whether I'm going to choose an E. coli or whether I'm going to choose an yeast, that depends on the product that I want to grow, but it also depends on how well they grow, right? So understanding that is very, very important when you're designing a bioprocess. Now, what is an efficient bioprocess? Whenever we talk about efficiency, efficiency has to be defined in terms of time. Efficiency can be defined in terms of money. If it is able to make you more money, it's an efficient process. If it is able to save you money, it is an efficient process. If it takes less time, makes you more product, it's an efficient process. Other than that, it also depends on the production of organism. It depends on the optimal conditions for the desired product. If you're using the optimal conditions for the desired product, you're going to have a very efficient bioprocess. The product value will define your efficiency of the bioprocess and the scale of production. If you're able to produce in mass, a grand scale or a large scale, you'll be able to make more money out of it, and your process would be an efficient process. Like setting up a startup, you should be able to make some money at the very end. So this includes, so when we are trying to do the efficiency calculation, we have to look into the size of the reactor, the type of the reactor, and the method of operation that we are going to use, which will give you the best for a given conversion. So if you're converting substrate A into product B, you should make the right choice in terms of choosing the right reactor, the right size of the reactor, right? A single person does not need an airplane. A lot of people will require an airplane, let's say. If I'm a billionaire, I will definitely need an airplane, but I'm just saying in terms of like as a single person, I might be happy with a bicycle to reach from point A to point B. So depending upon your need, you have to make the right choice in terms of choosing the right reactor, the type of the reactor, the size of the reactor, because all of that will define how energy efficient your process would be. If you're choosing a bigger reactor for a smaller reaction or for a smaller product size, your efficiency is less. Okay. Now, the bioreactor design, there are a couple of other things that are involved in it. So those will be your control and the positively influence the biological reactions, and you have to prevent contamination. The capital investment and the operating cost, we already discussed that. So during fermentation, maintenance of monoseptic condition is very, very important. So anything and everything that is added inside is sterile. So the nutrients that are going in are sterile. So you're only growing one microbe inside the microbe that you have put in, the culture that you have put in is the only thing that is growing inside the reactor. You're not adding any contaminant to it. You're not adding any other microbial colonies to it or any other microbial population to it. So optimal mixing with low and uniform shear rate. Now, why do we need optimal mixing in terms of a bioreactor, and why do we need a uniform shear rate? Optimal mixing will help to homogeneously distribute the nutrient and oxygen if you are sparging oxygen. Uniform shear rate, if you are able to identify the optimum shear rate or optimal movement of the sparger or the rotational speed of the sparger, the mixing would be ideal. You're not going to break your cells. If you make your not the spark jets, your impeller. If you make your impeller to run fast, you might break your cells. That will lead to cell lysis. Then comes the aeration, surface or direct sparging or indirect. So depending upon what will provide you the best and optimum O2 distribution within the bioreactor, if it is an aerobic, if it requires aerobic conditions, you need to identify the best way of providing that aeration into the bioreactor. So maintenance of adequate heat and mass transfer and the flow conditions. So let's say for a fed batch, we have to have an input. Let's say for CSTR, you have to have an input and an output. It has to be maintained in such a way. You cannot have a lower output, higher input, because then your reactor will overflow. Correct? So you have to maintain that optimal condition, the balance, so that your cell, the cultural growth or the cellular growth is maintained within the reactor at all times. The products and the byproducts removal is also there. So to maintain the optimal conditions within. Now bioreactor, for that we have to look at, like, it provides a controllable, so my mistake. So what does bioreactor do? Bioreactor provides you a controllable environment, enabling the biochemical and the biomechanical requirements to create a desirable product. So you are being provided with both the parameters to grow your product to the best of its capabilities. It enables close monitoring and control of the reaction parameters like internal and external mass transfer, heat transfer, fluid velocity, and shear stress. So one unit operation can provide you all of this control, and then you can grow your cell culture within that unit operation. This is a diagram of a typical bioreactor. The previous one that I had shown, that was a diagram for a typical bioprocess. This is a bioreactor. So what are the parts, and what are the most important parts for us would be your agitation system, that means your impellers. The design of your impeller will also be a parameter at one point of time. The sensors, the sensor probes would be for your pH and the temperature. So you're looking at your pH, you're looking at your temperature. What else can I use the sensors for? Dissolved oxygen, right? DO. What else? The level? No, let's say it's a batch reactor, so the level is maintained. So you're looking at the dissolved oxygen, you're looking at the pH, you're looking at the temperature, right? These are the major sensors. What else can be used? Or what else can I sense inside a bioreactor?
Conductivity.
Conductivity? Why? And in what sort of application will you use conductivity sensors?
Majorly conductivity measure by for insulin production, they measure the conductivity to consider how much salt concentration, how much is available in, or how much
If you
cells
change your salt concentration, that will change your pH also.
yes, but majorly in industrial level conductivity also one of the parameter that can
They do use conductivity meters, yes. The conductivity sensors are there. I personally think if they have been used when you're working with microwave fuel cells, mostly.
Cell counting sensors, NIR probes.
Have you ever seen
I have seen them.
a bioreactor having that?
Yeah, I have seen that.
So when they are doing the NIR probes, you said.
NIR probes. I don't know the full form, but they are used to measure the cell, like what is the cell count at particular time.
Okay. I haven't had that experience yet. So thank you. So there are a lot of many sensors now that we can use inside a bioreactor. So you have your reactor tank, that's a typical design of a bioreactor. You have your feeding pump, which is basically used to push in the nutrients inside. You have your spargers, which is going to provide you the aeration. You have your agitation system to maintain the homogeneous distribution of the nutrients and the oxygen. You have the thermal jacket. Now, this thermal jacket is to maintain the temperature within. Okay. So, this is a typical diagram. So, parts would be agitator, baffle. Now, baffles are sometimes here in the corners of the bioreactors. In this particular diagram, it was not there. Now, why do we add those baffles? To remove the vortex, right? So, because when you're running your when you're running your stirrer or your impeller, you generate the vortex. To break that vortex, you're going to use the baffles. So, you have your sparger, the cooling and heating jacket to control the temperature. You have your control systems. Typical ones would be temperature, pH, and dissolved oxygen. And thank you for the cell count part. I will add it. Now, let's get into the classifications of bioreactors. Now, bioreactors can be classified based on the presence of or absence of oxygen. Before we start discussing that, can you tell me the two types of bioreactors that we have actually discussed once on the day one? I asked you a question about if you have potato peels, or let's say you have orange peel, what sort? So either it would be a submerged bioreactor or a solid state, right? So that's also another classification. So whether you're going to have everything submerged inside or not. Then you have your in the presence of oxygen or in the absence of oxygen. So anaerobic fermentation, so no aeration is required. Initial preparation of inoculum may require aeration because you want to grow the cells. The gas released during the fermentation is sufficient to provide the mixing. So when you are in anaerobic production, you will have some gas generation within the reactor, inside the reactor as the cells are growing, and that gas bubble is going to move up. As it is going to move up, it is going to make the mixing of the nutrients with the cell culture. The product recovery may also require anaerobic conditions. Majority of the time, if I remember correctly, in the lab we provide anaerobic condition by purging with nitrogen. For aerobic fermentation, you have non-stirred aerated reactors, which would be airlift bubble column reactors, in which you're basically, so there is no impeller, so it's mostly being mixed with the aeration. Stirred and aerated reactors, where you have the stirred tank reactors. So CSTR would be a part of that. Some of the batch reactors would be part of that. Fed batch are also part of that. So on the right hand side, you can see an example of airlift bioreactors, where you have the draft tube internal loop configuration, a split cylinder device, and then you also have an external loop system. So you'll see majority of these things are moving. A very similar design for this would be, if you have learned dehydration of food products, there is a design of one of the dryer like that. What is the name of the dryer? Where your product is fluidized. We call it fluidized bed dryer, right? So it's because you want to make sure that your air is uniformly distributed. Your product will dry faster in that case, right? It's very simple, similar concept applied here, or this concept was applied there. Now, based on the modes of operation, now this is what we have been discussing. I have actually stated these names multiple times. So it can be a batch reactor, it can be a fed batch, or a continuous stirred tank reactor. Now, when it is a batch reactor, no input, no output. So it's like a pressure cooker. You put everything inside, close it, let it run. Once it's done, gives you the signal, take your product out. Okay. In a fed batch, we can add the nutrients. There is no output, but we keep on adding the nutrients. So, the intent of a fed batch would be to reach the maximum cell mass, right? We keep on adding, the cell will keep on growing. The cells will grow continuously till the time all the substrate that is present inside the bioreactor is consumed. If you keep on adding the substrate, the cell mass will keep on growing. So, the intent of the fed batch reactor can be to have the maximum cell mass. So, now from your own understanding and experience that we have had through conversation in the past couple of days, where will you use a fed batch? If the product is intracellular, right, developed intracellularly, you want to have the maximum biomass that you can take out, crush, and take your product out. That can be one application. Where else will you use it? Will I like to use it in a, let's say if the product was extracellular and the cell can continuously produce it? In that case, I can go for a continuous stir tank reactor, right? So, depending upon the type of product that you are going to develop, you can choose the right type of reactor, right? So, in the continuous stir tank reactor, you have your S0 and X0. Your S0 is basically your substrate concentration, your substrate at time T0, that is initial substrate concentration or the substrate that is coming inside. Then you have your X0, that is your cell biomass concentration at time t is equal to 0, your initial cell biomass concentration. X, S and P are your, X would be your cell at the end of what is coming out, S would be the substrate that is coming out, and P is the product. Now, in a continuous stir tank reactor, if you see the output, you have your cell, you have your substrate, and you have your product, correct? You still have your substrate. So, not 100% of the substrate has been used. The cell that is coming out is going to reduce the cell count inside. So, you can do a recycle in that case. So, that is the reason in CSTR we generally have a recycle. So, what are the design considerations for a bioreactor? The most important for any startup in the world, capital. Similarly, if you want to set up a bioreactor facility or any fermentation facility, the very first thing that will come into picture would be your initial capital expenditure. Now, out of all these three reactors, which one do you think is the cheapest? Batch, because of ease of operation. Or you can buy as big as you want, you can put as much of your substrate into that, let it run, and once it is done, open it, take your product out, right? So, substrate, if you see, that's why I'm saying, like, if let's say I come to you and say that, okay, I need this particular product, your choice of reactor will depend on many things. One of the things is how much money I'm going to give you as your initial capital, right? So, your initial capital expenditure will define the type of reactor that you're going to choose. The ease of operation would be the second most important parameter. Less downtime. Now, what is your downtime? What is downtime? So, you have a reactor, all the reactions are done. So, let's say it took, you started it now, it took 3 or 4 hours for the whole process to end. After 4 hours, you open it, you remove your mass, cell mass, you clean it. It takes you 2 hours to make it ready for the next batch. The downtime is 2 hours, right? Runtime 4 hours. Okay, so we'll be using these terms like downtime, the T, and the runtime or the batch time would be your 4 hours. Downtime would be your 2 hours. So, TB would define your batch time, TD would be defining your downtime. So using these parameters, you will be able to understand the efficiency of your process that you are working with, or even the productivity. You can define productivity not just on the terms of how much cell mass you can produce or how much product you can produce. You are also going to look at the total. Like if you're running your reactor every day for 365 days of the year with a specific downtime in between, your productivity will depend on how many times you have ran it, how much of the downtime you have had in between, because that will define your total time. So total time is batch time plus TD, simple. So how, if I tell you like you are able to run 2 batches a day, if you're running your system for 365 days, how many batches did you run? 365 into 2. And that will give you the total mass you would have accumulated, right, over the year, or what your yield had been over the year. Maintenance, ease of maintenance, not just maintenance, ease of maintenance is also a very, very important parameter when you're trying to choose a bioreactor or even design a bioreactor. Skill requirement, there was a question, I remember. So skill level that is required to run a bioreactor, depending upon the choice of bioreactor, will differ. If you add too many complexities, you need somebody who's highly skilled, right? If you have less complexities, you can run it without any skill. Give me one very good example of a batch reactor, not a continuous stirred tank reactor, or let's say any of the reactors which have been operated by least skilled people, a product that was developed by least skilled people. They don't have to have a degree in engineering. Come on, you do that every day at home. Your mom does that. Yogurt, curd. Another in North America, it would be moonshine. Have you heard about moonshine? Illegal liquor,
Okay.
which you can do brewing in a jungle. You don't need a highly skilled engineer to be in the jungle, hiding from the police, brewing your fermentable alcohol. That's your moonshine. In North, at one point of time, it used to be illegal to actually brew moonshine, and people were still doing it. So that's a very good example. You don't need to have skill. You need to understand how the process works, right? Skill is if you can operate it. Technical skill is a different thing. So when you start adding complexities where you have to estimate how much of dissolved oxygen is there, are you able to maintain it? You have to do some calculations. Yes, technical skill is required at that time. That might change the type of reactor or the choice of reactor for you. If your choice is to make sure that a specific skilled level is utilized or employed for a process. The other would be your control systems, the type of controls that you would like to put. If you only require a basic control, that means temperature, what is happening inside, or you need your temperature, you need your pH, you need your dissolved oxygen, you need to know your cell concentration. You need to add all of those controls to design the bioreactor. Then you have your quality or the culture. The design consideration will require the quality of culture. The quality of culture matters a lot. What do I mean by the culture? So if you remember when we were discussing about the process to reach the reactor stage, we have to grow the cells in the lab. So we use the flasks, we grow in them in that. But then we are what are we trying to do? We are trying to grow the culture or the cells in the nutrient-rich environment, right? So that their metabolic machinery within the cell has aligned to that nutrient. But that nutrient has to be similar to the nutrients that they are going to get in the reactor. Might not be in the same concentration, but they will be getting in the reactor. So that small scale, pilot scale, large scale, that's how it goes. So you have to have that chain. You develop the culture, you build the culture, then you pass on the culture inside. Correct? Now that machinery, the metabolic machinery which is within the cell, that will keep on, that will change if you change the carbon sources. So let's say if I have given a carbon source 1 to the cell culture in the lab, and in the fermenter I am giving it a carbon source 2, what will happen? Will my cells grow? Yes or no? If that carbon source can be used by the cells, it might grow. If that carbon source is not to be used by my cells, they will not grow, they will die. The quality of culture will also make a lot of impact, because if you have an older culture, again an example would be your curd and yogurts at home. If you use an older culture, you might not get a proper set curd. If you are using a new culture, very new culture, where you don't have enough microbes, you might not get a proper set curd. But you have to have the appropriate optimum culture to grow in the fermenter system, right, or in the fermentation process. When do you reach it? How do you reach it? It has to be done outside the fermentation process. Okay. Now, the classifications of bioreactors, you have your solid state bioreactors, the cultivations in the absence of free water, of pretreatment of the substrate, and difficult to control process parameters. Now, solid state bioreactors, the cultivation is in the absence of free water. Pretreatment of substrate and is required, and difficult to control process parameters are involved in this. It's not as simple as regular submerged bioreactors like a batch process or a fed batch or a controlled stir tank reactor. Application of these are mostly for antibiotics, food additives, biocontrol agents, and bioremediation. In my research group, we have used it for food additives, specifically to get natural pigments. Natural pigments would be your natural colors from the food waste products. So we were using onion peels. We have used carrot. In North America, you get here you have your carrots with the top, then you have your leaves, right? Excuse my drawing. When you go to the market, you can buy the whole carrot here. There, they will cut this part and this part. And what you get is in between, and bunch of that. So this goes as a waste with the leaf. We call it the crown and the tip. The crown and tip is thrown away. So my research group, we mash it, we make a pulp out of it, put it in a solid state reactor, run it just to see if we are able to extract the carotenoid, whatever is present in it. Now, carotenoid is a natural pigment that will give you the natural orange color, right? Now, other classifications of bioreactors are immobilized cell bioreactors and the fluidized bed bioreactors. We did discuss about the fluidized bed bioreactors a little bit. Immobilized cells bioreactors are where reutilizations of cells is required. So your cells are immobilized into a different chamber, or you can say a flow cells, a flow cell, and then you have your substrate being pumped into that flow cell, and as it moves through the chamber, your cells are interacting with the substrate. Your product is formed, it is pumped back, and the substrate, whatever conversion has taken place, is pumped back. So it reduces the contact time, it increases the volumetric productivity. The fluidized bed reactors, you have your heat and mass transfer is efficient in this case because everything is fluidized. So the transfer of temperature, the transfer of or the mass movement would be appropriate. effective mixing between liquid, solids, and gaseous phases, and it has a very low shear rate, suitable for plant cells and mammalian cells. So depending upon the type of host, so let's say if you are going to use mammalian cells as a host to grow something, you would like to go for a fluidized bed bioreactor. Okay. Now comes the main aspect of your bioreactor design and analysis. So what are a couple of parameters that we have to discuss here? Sorry. So for the bioreactor design and analysis, till now we have talked about batch reactor, fed batch reactor, continuous stirred tank reactor, fluidized bed reactor, immobilized bed reactor. There is another type of bioreactor. Have you heard of this? Plug flow reactors. You know what a plug flow reactor is? Those who know it, please help me out. Okay. So in the chamber, do you have the cells immobilized? Or what do you have in the chamber? Because that's a plug flow reactor, right? So you have passed, in my understanding, it is you're passing the substrate as a plug. So it is moving from one side to the other side. It will get converted, or the substrate will get consumed and will go away, right? The product will be formed and that can come out. So this particular chamber in a plug flow reactor, does that have your immobilized cells? Yes or no? In this particular design that I have made, yes. So in a plug flow reactor, that is one way of passing it. It's a really nice design, but to be honest, I don't remember what exactly we use it for. I forgot. Dr. Bala?
Type of furnace reactor without any axial mixing.
Yes, there is no axial mixing, axial mixing. So in the axis, there is no mixing. It's moving from one direction to the other direction, right? Okay. So that's one other. So plug flow reactors. That's another type of reactor which is there. Fluidized bed we have done. Immobilized cells bioreactors we have done. Okay, so bioreactor design analysis. So what size of reactor, type of reactor, or method of operations are best for a given product formation? That's the very first question that you're going to ask yourself when you're trying to choose the type of bioreactor for your own process. Now it depends. It very much depends for your own project, sorry, not project, for your own case studies, depending upon the type of product that I have asked you to make, you have to choose the right bioreactor. But that choice will depend on the host that you have chosen, correct? It will also depend on whether your product which is made is extracellular or intracellular. What else would be there? The host, the product, you need aeration or you don't need aeration. Do you need mixing, continuous mixing? What else would be there? Somebody give the answer. Environmental conditions. Okay, process parameters. What else would be there? Will the amount of product that you want to develop be a condition? That should be the primary condition, the productivity, right? If you're having difficulty, go back to this particular slide. Ease of operation, less downtime, product variability should be less. Productivity or yield is also a very important parameter in choosing the right reactor for your process. Maintenance is another thing. The skill requirements, don't consider that for your own case studies. That won't be necessary. Control systems, yes, would be very, very important. The type of feedstock that you are going to use will also define. Now, how do you define the performance? At the very beginning of our lecture, we talked about from the monetary perspective and the time perspective, right? If you have to say if a particular process is efficient enough, I will say how quickly I was able to get my product, or how much product I was able to get to make the money out of it. When you are looking at from the reactor perspective, you will be looking at the reaction time. So how much time it takes for the conversion of the substrate into the product. So it is a measure of processing rate in a batch reactor. And so whenever we are working with a batch reactor, the performance measurement is based on the reaction time. When we are working with, let's say, a flow reactor, like a CSTR, continuous stirred tank reactor, we are looking at space time and space velocity. So space time and space velocity. So space time is the time required to process one reactor volume of feed, and the space velocity is the number of reactor volume treated in unit time to define the performance of a flow reactor. So both space time and space velocity are used to define the performance of a flow reactor. Reaction time is only used to define the performance of a batch reactor. Before we go into the next set of slides, which will be later, I just wanted to go back on a few slides and have a bit of a discussion with respect to your case studies. That was the intent. Now, when we are looking at your case studies, so case study 1 was monolin. Please correct me if the spelling is correct or not. And the case study 2 was alpha amylase. For monolin and alpha amylase, for monolin, it's a plant protein. It's a protein as a product. Now, what will impact the quality of the protein or the product in your fermentation system? Your pH will impact it, the temperature will impact it, so the process parameters, right? So you have to be very careful in terms of what reactor you will choose depending upon what will happen to my product at the very end. So you want where you have maintained the temperature, okay? pH will change based on if the product is going to add to the medium. If my product is acidic or basic, that is going to change the medium pH. The protein, if that is extracellular, is it going to change the pH of the medium? Guess, yes or no? So when you add protein to a media, will the pH of the media change? Or any, let's say if you add protein in the water, does the water pH changes? You have your protein shakes, right? If you go to the gym, does the pH of your protein shakes change when you mix water in the powder protein? So let's say it doesn't. Let's say it doesn't. I'm not agreeing with this or disagreeing with it. Just for the sake of this conversation, I'm saying let's say it doesn't change the pH of your medium. The only parameter that remains would be temperature, right? If the temperature is also optimum, your interest is that you get maximum amount of protein generated, right? Then you can decide what type of bioreactor it would be. Be careful in that. In case study 2, alpha amylase, that's an enzyme. Again, a protein. Whether it is produced extracellularly or intracellularly, depending on the host that you choose, depending on the process that you choose, will define the type of reactor you are going to use. So you have to be very, very careful in choosing the right reactor design for these type of processes. The second part of our presentation is pretty long. The fed batch and the batch reactor, and this part was not too long, so I would like to end the session here for this one, and we'll meet again at 2 to start the second session.